So, I would like to introduce the Professor John Girkin, professor at Durham University, the United Kingdom, and director of the Biophysical Science Institute.
Right, thank you very much. So what I'm going to try and do this afternoon, one thing I'm definitely going to do is to build on what I talked about in the previous two talks. And remember that if there was one take-home message I gave from the first talk, it was reiterated today by Bruce, which said, you've got to have contrast. But I'm also here going to look at how you can improve the resolution. So once you've put a material or there is something that you're looking at that's giving you optical contrast, how can you improve that? And I'm going to try and teach and give you an idea, even for the biologists, the chemists, and the clinicians that might be present, two fairly difficult concepts in physics. So we're going to learn some quantum mechanics, and we're going to learn some Fourier theory. But hopefully I'm going to do this as fun. And then at the end, if you remember, in the last talk, I set you a challenge. And I do want some answers. So what I'm going to be talking about is this idea that can you beat this diffraction limit, the smallest that you can focus light down to? Can we actually image better? So we know that that's limited partly by the wavelength of the light that we use, but can we use an optical imaging system to measure or look at structure that's a few nanometers in size? So this diffraction theory, if you like, was generated by Abbe, and this is his memorial, so can we beat him? I also thought I should put up some very modern, up-to-date references for this work, because you'll all want to do this. So this is 1873, so these are really papers that you should go and get and look at regularly. These are obviously by Abbe. This is an interesting person, which shows perhaps how science was done 150 years ago. That person got the top mark in mathematics at Cambridge in the year that he was there. And he then just worked as an electrical engineer or an engineer, and in his spare time he worked on diffraction theory. So he's known as a first wrangler. It meant he came top of the whole maths exam in all of Cambridge, and he became an engineer and worked on this in his spare time, a gentleman scientist. So how small can we focus light? And this is going to be true. In fact, what I'm about to show you is true not just of light. In any optical system, projecting system, optical tweezers, anything that uses light or in particular uses waves for looking at things, what's the smallest? Can anyone remember what the important things are that we need to consider? Can anyone remember the equation that I gave you? So what's the answer? Can anyone remember? Sorry? The numerical aperture, good, we're going to come back to that, and the wavelength. They're the two things, so the size of the lens, and you'll see where that comes around to. But there's actually different ways we can look at this as well. So the normal answer, they all relate to the wavelength, the aperture, and the focal length, and if you put those two together, that gives you the numerical aperture. They're all based on some approximation. So if you focus the light down, you get a series of concentric rings, and we just say, well, we want to look at the light that's in the first ring. And that's what gives you this. If you look at the peaks, so this is using what's called the Rayleigh Criteria. So we've got the focal length of the lens, the size of the lens, if you like, here, and the wavelength. And if you turn that into the numerical aperture, it's what I gave you before. So good, people remember that from before. You can actually, so if you remember, this is what we get with a single, nicely focused one. If we've got two close together, these light is beginning to overlap each other. That one we could resolve, that one we can't resolve. And we come up with this formula. We can do a similar thing. So if you take a laser beam out of most lasers, not laser diodes, I stress in this case, they form a Gaussian beam. It doesn't matter what a Gaussian beam is, but you can actually work out what the size should be for a Gaussian beam. And again, we see in essence the same thing, the focal length, the size of the beam at the lens, and the wavelength. They're all coming out to be similar sort of things. Little terms in front, 0.6 or 0.5 may change, but they're all about the same. But what's the ultimate limit? What's the smallest we could possibly focus anything down to? There are tricks, and I'll show you some of the tricks in a moment. So there's a technique called near-field optical microscopy. So you can imagine a very, very small aperture which produces lots of, and I'll explain this a little bit later, high spatial. So you can use a very, very small hole, and that is one way, but you don't get much light through a very small hole. And the other thing is that the electric field, the light field that's being produced through this small hole disappears very rapidly. There is also the idea, so we've talked, people have talked previously about the refractive index, the speed or a measure of the speed of the light through the material. It's actually possible to make materials that have what's so-called negative refractive index. And in fact, if you do that, you can make perfect lenses which actually can, in theory, focus things down to very, very small spots. The other use for negative refractive index is for cloaking devices, for spaceships. So the Romulans have developed this as a technique. As of yet, I don't think the Enterprise had a cloaking device. But this is also a way that you can bend light around objects. My quantum mechanics. So some of you in the audience will have come up with this Heisenberg's uncertainty principle. What's it mean? And I promise you there is not lots of maths. This is all my physics, which is very hand-wavy physics. So what it says, that x is a position and p is the momentum. So momentum of something is its mass, its weight, times by the velocity, the speed, and the direction that it's moving in. And basically, if you measure what this says, if we make delta x, if we measure the position of something more and more accurately, its measurement of, if we make this smaller and smaller, this has to get bigger and bigger, because these two terms here can never get smaller than that. H bar is Planck's constant. It's just a number. Don't worry about it. But it shows that if we can't measure the position and the momentum of the light simultaneously with absolute perfection. There are other pairs that go in here, energy, and can be looked at. So, and what we can say is that this x, the position, so now think of light as a particle. So it's a photon, it's traveling through. And so as I send particles through a lens, they're going to go in slightly different directions. So if I then look at that, the distribution of where these particles land, and I can use some statistics and call it the standard deviation. So in theory, they'd all go dead straight, but some of them are going to go in slightly different directions. And then we can do the same thing, as you'll see in a moment, for the momentum. And there's also, again, invented, discovered about the same time, another scientist, Louis de Broglie, who was a Frenchman. He actually came up and actually said you can relate the momentum with h bar, and actually k here, and you'll see why I've used it later. It just means the wavelength. So we can relate the momentum of the light with its wavelength. So this says we can't measure two variables perfectly, and this one says we can relate the momentum to the wavelength, in this case, of light. So you're going, so what? I've given a clue in a moment. You can think a little bit about these photons going through a lens and ending up in different places. And that's going to be its position. So we can think of the probability of where a photon might go as, and you'll see in a moment. And the other one, if we've got a circular beam, we can say that the standard deviation is the measure of the size of that beam. So, what's going to happen? I'm going to have a cylindrical optic. I'm going to take a normal lens. What's this actually going to do? So here we go. This is for a lens. So this is my distribution of photons. So if you like, it's the intensity profile that I have. So if a beam is highly intense, it has lots of photons. So in the middle of this beam, I've got more photons than on the outside. So it goes through the lens, and the distribution changes. We focus them in. And this is just the focal distance down here. And we can look at this in a moment. So we know from that little thing that I had before, if we look at the photons here, and if you remember, x and p were multiplied together, and we add h bar over 2. So we can look here, so we can say that with the momentum of the photons here and how they relate to their position across here. It's just by uncertainty. And we can also then say that theta, so this is the angle that they're going on, well, that's going to be related to the velocity. So that's the speed and the direction that the light is going in. And we know that momentum is the mass times its speed times its velocity. So we can actually work out what the angle is. And so then we can just put that bit in there. And this divergence, so there's a thing in there which actually says how much it's going to spread. So if light was perfect and it traveled in straight lines as you draw with a ruler, you'd get a point here that was infinitely small. We can't get something that's infinitely small, because if it was positionally that accurate, its momentum would be spread around, i.e. its velocity, so the light particles are going to go to different places. And we can actually look here, so we can put our k back into it, and we can make the assumption that put that back into there. And then now for a focal lens, we said it's gone f down here. It's got this size here, so we can work out what the theta is for a lens. And we can put those bits together. And what we end up with is this thing here, that if we take its size, or the distribution of photons across here, and the distribution of the photons across here, it's again related to the focal length of the lens, and the wavelength of the light comes into that as well. So it's the focal length, the wavelength multiplied together, and then if we wanted to find the spot size down here, we'd put that x underneath there. So in fact, and this is going to be true for anything that we're looking at. So in fact, these theories, this is just, if you like, it's a little bit of fun. Some of you may not have thought that it was fun, but it's a different way that you can think of light. You don't have to think of it as a wave. We've talked about it being photons, and this is just a different way by looking what the chances of a photon being in any particular place. So that's set an ultimate limit. What are the ways that we can actually go about cheating, partly cheating what's on there? Well, you'll see in a moment I'm going to talk about what's called turf, stead, storm, and structured illumination, adding something to what I said the other day. So basically, if you want to do any of these sub-diffraction limited things, what you need to do, first of all, is think of a good name, a good acronym, and then you worry what the optics are going to do. So the first thing is come up with a good name. So you have storm and palm, and there's a whole lots of variations that all go together. But how do they go about beating that limit we've just looked at? Sorry, there's going to be a little bit more physics here. So this total internal reflection microscopy is based on total internal reflection. And it's an evanescent wave, and we talked yesterday about plasmons. We don't have to worry about that, but it can work in reflection or fluorescence. So what we've got is a glass block. And up here, we may have something with a lower refractive index. It could be air, it could be water. And we put some light in, and most of the time it goes straight through. It's what you'd expect. But if you hit light at the right angle, and it depends on the refractive index here and the refractive index here, it gets completely reflected. So in fact, this is an example that I use in the lab. This is just water. We put some laser light down it, and you can see it's being bounced down the water. Makes a nice demonstration, because you can put a big bowl underneath, put some dye in it, and when you get the angle right and the water flow right, you get total internal reflection, and the whole of the bowl starts to glow because the light is being guided down the water. This is exactly the same way an optical fiber works. But what's going to happen right at the surface here? And let's say for the moment that we put something on the surface. Let's say it's a cell. Now, in fact, there is some of the light wave, some of the electric field that makes up the light leaks out a little bit at this point. And the amount that it leaks out will depend exactly on the refractive index. So some of the energy from the light could come out through the sample. And this is exactly what happens. Now, as I said, if you remembered in one of the earlier things, this electric field from the light falls off very quickly. So it's not as if there's going to be a strong beam of light normally coming out through here, but the electric field is alternating. So it's as if the light here is starting to stimulate or to drive up, in the case of fluorescence, something that happens. So this is another way of looking at it. It looks more complicated, but we put laser light in, it's bounced along and comes out through here. But what we've got are these so-called evanescent waves. It's some of the electric field from the light, if you like, leaks through, and those are sufficient to excite up fluorescent molecules. But this electric field dies away very quickly. So it's only typically between 10 and probably 100 nanometers. So what we can do is we can design this. We can either put laser light in from the side and look to what fluorescence is generated in this place, or we can do it in another way. You can use a special microscope objective which has exactly the same effect. So you put light in down the side at a very steep angle, and then you look down the middle of it, and that's what happens here. So the light is coming in so steeply through this objective lens, it couples and the light gets totally internally reflected. If there's a cell here, we generate some fluorescence which comes down in this direction because the fluorescence goes in all directions. But this excitation is only going to happen in a very, very thin layer. So this is an example. I think this one was by Peter So from MIT. So this is what the cell looks like normally in wide field fluorescence. But when we look at it in total internal reflection fluorescence, all we see is the cell, parts of the cell, so this is a single cell, that is in very, very close contact or just above the glass surface. So we actually have a way now of taking an optical section, a very, very thin slice, that's less than the diffraction limit. We've cheated a little bit. So the resolution in this axis is exactly the same as you'd expect for a normal microscope objective, but in the depth, we've produced a very, very thin slice. The largest depth you can go to is about 500 nanometers. Typically, it's about 100 nanometers. And this is an example of it being used. I apologize, I tried to find the original, but this is from a particular paper, but I've had to cut and paste it. So in this case, what we did is we put light at two different wavelengths in there. One is from a monochromator, which just did some normal fluorescence imaging, and this is a frequency doubled laser diode, which went in to excite the total internal reflection. The light came back through here and went onto a camera, which then went onto the computer. And in fact, in this experiment, we were doing electrophysiology, so we had an electrical probe going into the cell as well. And this is what it looks like. In one way, you've got just this small evanescent field from the laser, and you've got this large area that was produced. And this image has failed absolutely abysmally on the bottom right-hand corner. You'll just have to trust me. In fact, if you go on to the next slide, in fact, what you would see here is we were actually looking at calcium, small pinpoints of calcium within the cells. And it's very difficult to see here, but I think if we look at this one here, so this is the the bright image, the wide field fluorescence image, the colors being different intensities, and this is then just looking at the excitation that was taking place right just above the cell membrane. And we were trying to compare what was happening at the cell membrane with what was happening slightly deeper inside the cell. So this is a way, so this was giving our wide field image, and this is just looking at the first, in this case, about 150 nanometers inside the cell. And we actually electrically altered the cell at the same time, and we went to see for changes that were taking place. So this is a method now that I've got for taking really very, very thin slices. So how can we actually do it in some other ways? So that's taken a thin slice, but we'd like to get high resolution in the other axes as well. So there are basically two ways of doing it. One is we can see in a moment, we can localize single molecules, or the other way is we can use patterns of light, or we can get a non-linear response in the light. So the first one I'm going to choose to look at is this technique called Stead, which was primarily been driven forward by Stefan Hell in Germany, though it was invented at about the same time between him and someone working in a company which I only learnt about a year ago in America, who actually has a patent on it as well. And it stands for Stimulated Emission and Depletion Microscopy. And you're going, what on earth's that? Right, we'll come back to that in a second. It only works in fluorescence. I would make the comment it's not really been used on real biological samples yet, and I'll show why I say that in a moment. At the moment, I think this is the current record. He uses light, and he can image to 28 nanometers. So that's about, in this case, it's about 30 times better than you would expect to get for the diffraction limit. So how does stimulated emission work? So a bit more quantum mechanics, but this is pretty colored picture quantum mechanics. So these are the energy levels that are in here, and we know we put blue light in, lose a little bit of energy due to vibrational losses, and then what we get is fluorescence coming back down. And you go, well, so what? That takes around about, we were talking about it yesterday, in a typical fluorophore, as if there was such a thing, let's say it's about 5 nanoseconds. But you can actually do it a slightly different way. So let's excite it up here, and it loses a bit of energy. And what happens now is if a photon of that wavelength comes in, it goes across there, and it stimulates. So in other words, this atom, this electron is sitting up here, and it's thinking, well, I ought to jump down to the lower energy in a minute. And suddenly it sees a photon of exactly the right wavelength. It goes, oh, I could be like that. I want to be like my friend there. So it jumps down. So what you get are 2 photons coming out. It's the same principle that's used in a laser. This effect, I think it's on here, I hope, happens very, very quickly. So once this photon comes in, this one jumps out for all intensive purposes instantaneously. So this is the fluorescence lifetime. It's going down, instead of in nanoseconds, it's pretty well gone in, let's say, a picosecond or so. So how do we do it with Stead? Right, we excite fluorescence, we put blue light in. There we go, blue light. And that excites my nice fluorescence area. And then I put a second pulse of light in. And this light is delayed, it's a few picoseconds later, and this light is in the wavelength of the fluorescence. So in this case, in my example here, it's green light. So I put green light in. But I'm only going to put it in certain areas. And now what happens, as that green light goes through, it depletes, it stimulates the emission in that area. So if I then look a short while after, what I'm going to get in this very simple diagram is I'll have removed all the fluorescence from here and all the fluorescence from there. And therefore, what I will end up with is just this bit of fluorescence when I then look with my detector. So typically what happens, if we then put it in, rather than using a sort of straight light like that, what we tend to use is a beam, but it's a donut beam. So the beam that we actually put in is circular. In Britain, you can have a mint called a polo, which has a mint round the outside and it has a hole in the middle. I'm not sure what they're called in other parts of the world. So we actually put in a donut, that's the best example, a donut-shaped beam which has light round the outside, no light in the middle, and after we've put that pulse in, all we get left with is the little bit of fluorescence in the middle. And of course, this bit in the middle was in the black part of our beam. We had a donut beam with the hole in the middle. The hole in the middle has no light. Therefore, it can't diffract. So we can actually make that hole in the middle smaller and smaller and smaller. And what we then do is, by making it smaller, this bit in here, which then emits the light, can be smaller than the diffraction limit. So the way that it's done in reality is we have 2 lasers. You put the blue light in, that excites up the sample, and then what happens is we put, and this is done very simplistically, we put a light beam in with a little block in the middle, so the block means that we end up with a light beam that looks like that, which when we then turn that laser off, the only bit of fluorescence that left is the bit in the middle, which then goes back to a detector, and we only turn the detector on after we've put this light beam in. So we've now made we're looking from a very, very small spot, and then we can scan the sample around. And this is an example. This was taken, I haven't put the year on down at the bottom. I think it was 2009 from memory. apologize for not putting the year on. And this is a polymer. And it works very nicely in materials because you have to do this repeated cycle lots of times. So it's not always going to be very good for biology because you can't just do it once for each point. You have to go round several times. And of course, there's a risk then of either photo-bleaching or phototoxicity. But it works perfectly well for polymers. This is actually done with some mitochondria. So this is within here. The scale bar is 500 nanometers. So this is enabling you to look at individual mitochondria. The other problem is, it's a bit like the zebrafish imaging I talked about. You don't really want these things moving around too much, because if you've got to take if they're moving, it'll have moved before you take your next image. So there is a problem, which is why I made the comment, it's not used a huge amount on live samples. But you can get these images, so this is imaging little bits of the mitochondria within there at very, very high resolution. But you can also cheat in other ways. I can actually cheat by just using a normal camera. So if we have a round object, and I know that it's round, something like that, instead of saying, okay, the image is this size, I can do some mathematics on it and find the center of mass, right, the middle point of my object, and then you can say that that center of mass then forms the pixel that I'm going to build up in my image. Or you can do this, if you like, for movement. So I think this is an idea. So this is actually the movement of mitochondria that were having green fluorescent protein in. So what happened is these are the pixels on the camera, and we can actually look through. So we can image the mitochondria, which might say is 2 microns across, but we can spread that 2 microns out over a large area on the camera, and then find where the middle of that is, and then say, that's the middle bit, that's the point that I want to look at. And then in this case, what we did is we'd look and see where the mitochondria moved to. So this is the track of the mitochondria. And in this case, because you're finding the center of mass of the image, we can get down to about 1/10 of a pixel in the image, about 5 nanometers in the way this was done. There's slightly different ways of looking at it. There are actually other ways you can do it, and this is an interesting thought. So if I take an image of a thin piece of wire, it will be really bright in the middle, and let's say it's less bright at the outside because the wire's reflecting light. So if I then take that image and mathematically manipulate it and square the intensity, the contrast gets bigger because the bit in the middle squares and becomes even brighter compared to the bit at the edges. So it appears to become even thinner. And I can keep doing this. I can keep squaring it until I get noise in a problem. So if I keep multiplying my image, it gets brighter and brighter in the middle and apparently gets thinner and thinner. Is that really beating it? It's cheating a bit. The other one that I wanted to talk about before you start doing some work is storm and palm. And many of you may have heard about this. In essence, they're pretty much the same sort of thing. So what we have here is a photoactivatable fluorophore. So we've got a fluorescent compound that we can turn on or turn off with another photon. So here's my fluorescent compound, and it's not fluorescing at the moment. It's just sitting there. Even if I put light at the right wavelength, it won't fluoresce. So what I have to do is, in this example, I put an ultraviolet photon in, and it changes, if you like, one way of looking at it is it changes the shape of the molecule. And now it can fluoresce. So this molecule was turned off. It's now in a position that if it got light at the right wavelength, it will fluoresce. So here's my molecule now, and now I put some light in, let's say in this case blue. Now my particle will fluoresce. So it gets excited and it will emit a red photon. And it goes back into its ground state, and we can obviously then keep doing this, and it's just a normal fluorescent material. But it has one other property. Let's going through the cycle again. If I actually then put in another UV photon, I can turn that fluorophore off. So I can get it to change its shape again. This is not it's the principle I'm trying to get over here rather than the detail. So I've now got a fluorescent compound that I can turn on and off. And you're probably going to go, well, who cares? Oh, it's very nice. I can turn a compound on and off. So what we do in these two techniques here is we've got this thing. So we first of all put on a very small amount of ultraviolet light. And what that does is it turns on just a few molecules. And then we can just use conventional fluorescence imaging because we've turned on just a few molecules. But crucially, what we say is literally for each point of light that comes out, it's only come from one molecule, or perhaps two that are very close together, but let's say it's one molecule. So for that bright point, we can find the center of mass, find its brightest point, and then say that's where that molecule was. I'll show you in a moment. Then we can turn those molecules off, put some more ultraviolet light on, and then we can turn some other molecules on, just purely randomly. So STORM stands for stochastic. It's a purely random process. You just turn fluorophores on and off at different times. And clearly, if there are more fluorophores in one small area, there's more chance of them being turned on, so they will get brighter. So how does this look like? So let's say for the moment this is some tubulin or something that's inside a cell, some of the cytoskeleton. And what we do is we turn on just a few, we put some light on, and only these few molecules get turned on, and then we turn them off, and we do the same thing somewhere else, and then there, and then there. And then in the end, what we do is we just add up all these images, and then what we can produce is an image with all these crosses added together. And because we've found the center of mass of each one, we can make the assumption again that we can image to about 10 nanometers, because we're assuming that each fluorescent thing we see has only come from a single molecule. So the other thing I was going to say, which goes on to the last technique, is real information is carried in the detail. That's fairly obvious. And the detail is carried by high frequencies. Sorry, what's high frequency got to do with anything? So we can actually make any particular shape up of a series of waves. This is what's called Fourier Theory, and it was done for working out heat flow down a pipe by I think he was a French mathematician. So what do I mean by this? I can make a shape up, and what's this going to help us for? And I'll show you in a moment. So let's say we want to make a square. So this could be a physical object. It could be this advancer for the screen. So as I look at this, this is the intensity profile, if you like, of a sample of some description. So it doesn't matter whether this is a physical object or anything, but it's got nice, sharp, square edges. So how do I make that from a series of waves? How can I break that shape into lots of individual waves, each of which has a particular frequency? So if we just take for the moment the beginning bit of that, the middle bit of that, it looks like that. Oh, this is beginning to look possible. So if I then take this, this is what my square wave looked like, my square shape, and I've now fitted into it one wave, one sine wave. And you go, well, that's not very good. Sort of the same, but it's not really the same shape. So what can I do then? Well, in this case, and it's why I've chosen a very simple case, I could then say, what happens if I have a wavelength that now has 3 times that frequency? So the next wave has 3 times that frequency, and you think, oh, wait a moment, you see that bit there could fill in there, and this bit's going to take away a bit at the top, and that'll fill in there. Oh, that's good. I'm getting better. Still not very good. So let's add the next frequency. Let's add the 5th frequency. Oh, add those, but I'm doing pretty well now. That's not bad. So if you like, this information, and if I do it once more, and this is one, I've just got odd frequencies to 15, so it's only got, whatever's that, the odd numbers up to 15. That's getting not too bad. I should say, this is, if you did this purely mathematically, you can actually get it to be better than that, and I've exaggerated how badly this is at the top, actually. But you can see now that instead of having that shape we had like that, we're now beginning to get sharp edges and the sharp edges have the detail. If this is intensity, this is the point where we've got high contrast. So what use is this? How am I going to use this to improve my imaging? So let's think for a moment. I've got an object, and this is a very diagrammatic way of doing it. So here's my object, and outside the other side of the lens, and this does happen in optics, you have the plane where all these frequencies, so-called Fourier frequencies, are spread out. So if I take some image here, it goes through the lens. This is not exactly the way the light looks at. Comes the other side. So here is the center frequency, and here is frequency 1, frequency 2, and we can actually get, if we want to look at it in this way, we can look at minus f1 and minus f2. So these are the frequencies spread out here. But I'm losing detail because there's probably some frequencies out here, but they're not getting through my optics. They're not getting through my lens. So if I make my lens bigger, and now I've actually made its numerical aperture bigger, so I've made the lens bigger, I'm now going to gather these higher frequencies. So I can start getting, so I've now got frequency 1, 3, 5, and this was if it was a square wave again, and I can do the same thing down here. So I'm now getting more frequencies through. Oh, well, that's all right. Then I just need to make a lens that's infinitely big, and then I'd get everything I wanted to get from my object. Well, it's a bit difficult to make a lens that's infinitely big. So how's this going to help us to get more information? You've probably all seen moire fringes. It's when you take two patterns of lines and you cross them to each other, and what you see underneath is something. So this is an image which has a pattern, a shape inside it. And if we then put on top of that just another pattern of lines, it's not a single frequency. So I've now just put a grid on here of one particular frequency. And then I project that, and if I add those two bits together, what I see is a pattern. You can't really make it out here, but this is this pattern that emerges because we've put one frequency on top of the other, and it's been beating together, and what we end up with then is some detail that was apparently hidden in this original image. If you go back, you can actually just about see it. So what we've done is this pattern that was only just visible because it came in high frequency, it was high information, we can now begin to make it appear by putting on another frequency. So we've decoded the image. So how can we go about doing this? So what we can actually do, and again, I apologize, it's only the bottom one that shows up. It's just a piece of material. It doesn't matter what it is at all. I think it was just a piece of glass. But you might just be able to see that we've projected onto it some black and white lines. The black and white lines in this one should be running, I think they're running vertically, aren't they? Yes, they're actually running vertically, and in this one they're running at 90 degrees, they're running across that way. And this is mathematically what the thing looks like in this space called Fourier space, in the waves. And in fact, again, I apologize, it's not very clear. Right at the center of that, there is a bright spot, which is the zero frequency, but we've got on top of this grid. And if people want to look at it afterwards, that grid has produced a spot here and a spot there. So that's the particular frequency that's coming through in our image. And in this case, because the grid is at an opposite angle, the dots appear here and they're there. So how can we begin to use this? So if we project, therefore, a pattern onto our sample, the high frequency detail will beat with our pattern. It'll come through, and that means we can get it through our lens. Because the low frequency, the beat between the grid we're putting on and the structure now has a low frequency, so it comes through the lens. So this is GFP, it's tubulin, so it's inside the cells. And this is what it looks like. Again, this actually should have black lines. You can just about make it out. If I take the pen away, if you look here, it's continuous. Here, it's just about got some black lines on it. So we've taken an image with our grid on it, and we can then take that into our Fourier space. It's purely a mathematical thing. It doesn't matter. We just do it in a computer. and we can therefore produce one pattern. And what we can start to do then, in a very similar way to the way that Bruce talked about this morning, is we can move our grid around. And when it moves around, it will beat with different frequencies in our image. And we can move it around. So in this case, we've moved it in 3 different particular areas, and we've produced that. We can actually then rotate it as well as moving it in a number of different ways. And we can actually therefore get out of it 7 extra orders of frequency. So we've been beating it, it's then a low frequency, it comes through, and it's sort of hidden in here. And then by adding these extra frequencies together, because we know what frequency we've been beating it with, we can actually then reconstruct the image, take the grating out because it's in every one, and then what we end up with is this very high resolution image. So this is structured illumination imaging. So we put a pattern on, it beats with our thing, so the high frequencies come out as a beat frequency, which we can then detect. I'd ask you a question. Do these methods really beat Abbe? Well, we've sort of cheated a bit, because we're using, perhaps in the case of Stead, we're using time to get some extra information. In the case of the imaging, we're turning fluorophores on and off and guessing we've just turned on a single molecule. In the case of the last one, we haven't altered the resolution. We've just said, well, we can beat it and then do some mathematical fiddling, if you like, to get the image back together again. What is resolution? Again, really what you need to remember is you need contrast, and what sets the final limit. So hopefully that's given you some ideas, and it's put a little bit of background into terms that you may have seen, you may never use them, but there are these methods now, generally using fluorescence in different ways, the Stead, the turf, of beating the diffraction limit. Now, if you remember, I said that we've just built, or it's being built, a microscope that's basically 3 millimeters cubed. The actual final version, actually, this bit comes over here, but it doesn't matter for the moment. And it has an LED that's mounted, very, very small chip of an LED that's mounted in the back here. This is, as I said, it's 3 millimeters, so this part's about 1 millimeter. This surface is curved and reflecting, so it works, gives you an idea how it works. We can't alter the focal length. It's set at whatever we want it to be. So it's a fixed focal length. But it can be anything from contact to 0.5 millimeter, and the camera that's mounted down here can image up to 30 frames, and we can take the image off through a wireless thing. So it's very, very small. It just needs a small external battery. So my challenge to you was, what would you use it for? So has anyone actually been thinking about this, or have they been far too busy drinking caipirinhas and being comatose on the floor? Has anyone got a thought what you might want to use this? Anyone willing to volunteer an idea? I'm afraid you might have to end up running around. There's someone at the back that wants to. Yes, you need the microphone.
I had 2 ideas. One would be to monitor fetal development, possibly. I'm not a medical doctor, so I can't say anything about the safety of that. And the other is to monitor the growth of bacterial colonies for either medical or non-medical purposes.
Okay, so if you were going to do it in a fetus, could you do it in a human fetus?
Probably not, until it's been tested.
Why it's an interesting, why couldn't you do it in a human fetus? Would it not be easy to do?
Getting it inside the sac would be
actually,
difficult.
that's relatively straightforward. Okay. You just put it down an amniocentesis needle. It would really go down.
I'd also be worried about transmitting the information and how that would harm the development.
Actually, you could get the information out. The biggest problem is, what are you going to use as your contrast agent? So this is going to be looking at an area at most about 1 millimeter. The imaging area, you can get about 2 millimeters in the imaging area. I believe we're not allowed to genetically modify humans to get them to express GFP. I think there's a slight ethical issue.
If you looked at the difference between oxygenation and deoxygenation?
Okay. Yeah. So, for example, in that case, you remember, you'd be looking at sort of cellular resolution, and there is a reason, whether you'd actually want to embed it, particularly looking actually at the way mitochondria, the chromosomes separate in early cell development, is very important for in vitro fertilization. But you can't use a fluorophore, but you can use other contrast mechanisms. And yes, if it got further, you could use blood oxygenation. So in that case, there is actually just enough room to put 2 different wavelengths of LEDs on. So in that case, yep, we could do that and we could use it. Has anyone else got another idea? No? So you've got a 3mm microscope. Okay, yes, we've got someone over there. You've got to keep them fit.
Maybe look at the autofluorescence in bladder cancer and all these type of cancers, so that you can put it at the end of, at the tip of endoscope, and then use it to look at the autofluorescence to detect different type of cancers in the bladder specifically. Yeah,
I think that would be potentially, because again, this is giving, there's a good reason why you might, this is going to give you cellular resolution or subcellular resolution, depending exactly what focal length and exactly how you make the front lens. So potentially, you could insert this, you need a 3mm hole, but it's not bad, but you can get cellular resolution images from a thing that's only 3mm along. And yes, there may be a case that you could get autofluorescence. That works very well. And the other thing is, this is basically made of plastic, so the material it's made of is benign. So the thing that we're using it for, anyone else got any other ideas? I'll say what we've actually developed it for, and try and give you an idea of what needs to go into the way that you think these things through and the time it takes. So this is actually being designed to be work for imaging the kidney. Not in a human, but in a mouse model. The kidney in a mouse is about 3mm-ish. And in fact, what we think, we know we can get something very early on, it's very delicate surgery, but we can, the kidney sits in a capsule, and we can stretch the capsule very carefully and actually get this to sit very tightly on the kidney. And what we're trying to look at is the way the kidney develops, or the way it actually responds to high blood pressure. So it's actually related to cardiac disease. And the kidney, this has been genetically, or the mouse has been genetically modified so that only certain cells within the kidney actually produce any fluorescence. So it's been genetically manipulated with green fluorescent protein. This has taken about 2 years to get the genetic manipulation done and to get it working properly. So and that's actually quite quick. So sometimes you can do the genetic manipulation very quickly. There are ways of doing that. But in whole animals, it can become very difficult. And this is just to give you an idea that if you're developing some instrumentation, there are huge challenges in doing it in a clinic. Absolutely. And it's part of the reason why I've moved away from doing things in the clinic, because the time to get things in and tried was becoming increasingly difficult. But when you start doing things in animals and you have to do the genetic manipulation as well, that takes a long time. In Europe and in America, you have to get licenses for doing all this, and you can't operate, and the rules are very, very tight. So to get permission to actually put this inside an animal, also in our case, took about 9 months. And in the same time, we're developing the optics and pushing it along. So I'm using this to give you an idea of how long some of these things develop. I'm not sure, I will be completely honest, which is partly why I threw the question open to you, whether having a microscope that size is of huge clinical use. I don't know. Some of the clinicians here might like to make a comment, because remember, you're only looking at a very, very small area. So if you wanted to do a sort of big biopsy in the bladder, you'd have to keep moving it round and taking lots of images. So I'm not sure whether it's, but it will give you subcellular resolution. And as I was speaking, I think it was not last night, it was probably last night, to Colin, he was saying, yes, that still the gold standard for cancer or no cancer, to a lot of it, is histopathology. It's the shape of the cells. So it does give you that possibility. It doesn't give you, we can actually do a little bit about the depth, but not a huge amount. But I hope it's given you a thought of the sort of thing that you can think about doing. These cameras are routinely available. They're actually very cheap. They're about €100 each. The box for it is a little bit more expensive, and it just runs off through a USB thing, and we've actually made that even smaller. They're partly designed for endoscopes, but I'm not quite sure whether, I mean, nobody thought about doing it in a fluorescence thing. In this case here, this is the fluorescence filter that's put into it. So by now, you must be pretty tired of my voice. You've heard me for 3 of these talks. I hope these are some of the things that I thought we'd look at. Is it research or the clinic? Oh, apologies, it should be or. What's the specifications? So if you were actually going to do this in the kidney, and it was a question that was asked last time, what's the focal length, what's the numerical aperture? I'm not going to go into the detail, but you can begin to think what those are some of the things that, as an instrument designer, we have to worry about. So what resolution do we need? The timescale for the project. There are other considerations, the licensing as well. I think what I'm really coming for in any of these, think of the thinking process on imaging, generically. Crucially, before you start, what's the question you actually want to answer? And if you're working with a clinician, you don't always want to say, oh, I need to do this. Ask him really what's the information he wants, and then choose the most suitable tool. What do you want to see? And there's going to be a trade-off probably between resolution, depth, whether you can fluorescently label it or not. Does it need to be dynamic or static? Different methods. Clearly, the Stead system does not work on things that are dynamic. Actually, the structured illumination does, because you can move the grid around very quickly. Do you need 1D, 2D, 3D? How much information do you want? Is it going to be in vivo? And if it's in vivo, is it an animal? The other bit is, remember, in all of this, particularly if you're doing it clinically, we want to perturb the system as little as possible. You know, the ultimate perturbation is death, and clearly that's not a good idea in a clinic. You tend not to build up a good relationship. But even in animals, we want to keep it down as small as possible. We're sending light very often in areas that light it's not used to being there. So you can get all these other effects. So we want to keep that damage. We've heard several times today about the therapeutic effect and the non-therapeutic effect nearby. You need to get that balance right. I hope that with all this, you've appreciated a little bit about selecting the correct tool for the task required. And that's a generic comment. There's this idea of KISS. You know, this is a horrible thing that marketing people do, and you've seen the things where they wear microphones like this and dance around the stage. But it really is, choose the simplest method you can for doing it. And realize that there is a difference between a clinical and a research environment. Those of you that may be working in a research lab may not always appreciate the complications in a clinic, and vice versa. There's a difference. And I've had, at the end, I think my first slide said, I want to make you think. So have you had to think? I hope you have. And with that, I say goodbye from Durham for the moment. Thank you very much.
Thanks for a nice presentation. I have a doubt. Why you can't use Stead for the in vivo imaging and can use Storm or Palm?
Why can't you, or why can you?
Why can't you use Stead, but you can use Storm or Palm?
So one of the So one of the problems with using, I'm making a very big generalization here, and it has been used in some live things, but only specific. So one of the things is, to get the Stead, you can't just send it through that cycle once. You don't get enough signal. You need to send it 2 or 3 times in each small area. And then you need to scan that spot across the sample. And that means you're exciting and driving a fluorescent molecule around this cycle lots and lots of times. And there is a significant risk that it will break down and fall apart. So there are only certain fluorophores that work quite nicely with that. So that's one reason. The other thing is, if you're going, the scanning process is fairly slow, because I can only do it each 10 nanometer, 20 nanometer bit at a time. So if you think you're going to image a cell which is, and I can't do the sum in my head or straight off the top of my head, if you're going to image a cell which is 10 microns across, and each spot's 10, that means you'd need to do it 1,000 times. Yeah, got it. That's in one axis, so I need 1,000 spots there and 1,000. So it's going to be relatively slow. And if the thing is in vivo, there's a good chance it will have moved. With the storm and the palm, you can actually take images very, very rapidly. So you turn them on, turn them off, and you can do that very much more rapidly. So I think the fastest that somebody's done storm at the moment, I think they've done it at 30 frames a second. I stand to be corrected, but I think that's what the number is. I think Stead, the quickest anyone has imaged any sort of reasonable sized area, let's say a cell, has been several minutes. I'm willing to be corrected on that, but I think that's it. So it's mainly the fact that in a living thing, things are much more dynamic. And it's very, though I accept, that it's an exaggeration. It's a wonderful technique, and you actually get higher resolution. But there's probably a limitation. In fact, with Stead, the resolution is, in theory, infinitely small, because you can make the blackness smaller and smaller. Does that help?
It helps, but I was thinking that storm takes 10 of minutes to acquire the data.
It doesn't take. You can actually do it much more quickly than that. And I think the fastest one is now 30 frames a second. So there was one done by Susan Cox, who's now at King's College London. She modifies it slightly. Actually, she uses the natural blink of the natural fluorescence going on and off at slightly different times, streams a video on a very sensitive camera, and I think her storm images, or the equivalent, are done at around about 30 frames a second when she puts it together.
Okay, thanks.
Hello. So I just have a small question about resolution. So you briefly mentioned the deconvolution of the point spread function. So what's the best resolution that can be achieved using that method?
Sorry, what's the best way of doing deconvolution?
No, what's the best resolution that can be achieved using the deconvolution method?
So what deconvolution means is that you know what shape your spot is that's focused down, so you can mathematically remove that from your image and improve it. I think typically using deconvolution, what it's really used for is removing the out of focus. It's helpful for removing the aberrations. Because the crucial bit is when you deconvolve it, you can't add any information. So the high frequencies, the detail in the image, is not there. It hasn't got through the optics. So these techniques need to cheat to get that extra information through by having a pattern that beats against another one. What you can do with deconvolution, and it's a bit like my comment that I made, I can square the image, make it brighter and brighter in the middle. I'm throwing away some of the information, so I can improve the contrast perhaps because I've removed my instrument function, but I haven't actually added any new detail to it. I've basically removed the blurriness rather than adding detail to it by doing deconvolution, simply because the information is not in my image in the first place. Does that make sense? So if you want a number, what it can help for deconvolution, if you're doing confocal microscopy and you go deeper into it, you know your spots got bigger. So you can actually say, well, I can deconvolve that, I can pull out the instrument function and say that's what it should be. But you then end up with the optical limit in effect of the thing. There is a part that says you may get a root 2 improvement, but that's the best possible that you can do. But it's not really given you more information, because the information isn't in there in the first place.
So you mean to say that we don't get any more information and it's only a root 2 improvement over?
Yeah, the root 2
whatever.
comes about because you get a level of confocality, you get a level of coherence to it in some cases if you do it in confocal imaging instruments. But so basically deconvolution helps you remove noise or artifacts. It can't give you more information because you haven't got it there in the first place.
Thank you.
So the structured mask that you showed for structure illumination, it looked like to me its orientation shifted.
Yes, that's right. So you beat it at different frequencies with different components, yeah.
Right, which that's how I was taught structure illumination. I understand how that works. But I've seen in some of these presentations and elsewhere where it'll be a structured mask that's just shifted laterally. Yep. And I assume that you lose some of the phase orientation information that way, and I'm not really sure how this works, unless it just gets rid of some of the or only helps axially?
So the hand-wavy answer to that question, and it's a hand-wavy answer,
Okay.
is that if you, so the structured illumination I did here, which looks at, this is the way of thinking about it, beats with lots of different frequencies at different angles. And that's how you get some extra information back. If you just move so it's the I'm going to get it wrong. The opti-grid is the Zeiss one and the optitope. There's two of them. I forget which one.
Abitone or something
Thank
like
you.
that.
Yeah. I'll tell you a classic patent error in a moment. That moves the grid that way. And what they do is take 3 images. Yes, it's just moved horizontally. And the hand-wavy answer to that is, in the focal plane of the microscope, that illumination adds up to give you, after 3 movements, even illumination. Outside the focal plane, it adds up to give you nothing. That's the simple way of looking at it. If I don't want to go into the maths, I can show it to you if you like, or if you're interested, if you look at the original paper by Neil but that's the hand wavy. So if you just move it 3 times, think of it as saying, right, in the focal plane it adds up to give me even illumination. Outside that focal plane, when I do the manipulation with I1 minus I2, all squared, I should add, they give you nothing above and below. The classic patent error, if you ever write a patent, so Mark Neal and Tony Wilson patented the grid pattern, and I think they sold the rights to that to Zeiss. I think that's the right one. And the other grid came out short while afterwards, and they did that. And the patent in one of them says move it horizontally, and the company got round it by moving it vertically, and that got it through the patent. So it's a classic. I have another classic that I made a mistake with LEDs. Wow. It's just an example when you write a patent. If they just said is moved, but the patent says was moved horizontally. Classic patent error, if you want to view it that way. But so think of it as the way, in one plane it adds up to give you even illumination, outside it adds up to give you nothing when you do the correct mathematical fiddling.
Thank you.
More questions?
Again, if anyone wants to come up, I've given this in a very hand-wavy way. I've deliberately not done the detailed maths, and if you didn't follow what was happening with the uncertainty principle, don't worry about it. It was just a bit of fun for people that may be aware, and how you can apply things from slightly different directions to solve problems occasionally. It's not important if you didn't understand it. The Fourier one, I hope you'd get a handle on.
Thanks again.
Thank you very much.